welcome to Weekly Infusion with Dr. Drew Pinsky and Dr. Bruce Heischober. Weekly Infusion addresses medically related topics. It's in the know, entertaining, and everything you want to know about health and medicine. Now may I present to you the very wonderful Dr. Drew and Dr. Bruce.
Hey, welcome to another Weekly Infusion. Bruce, very exciting.
Good to be here.
Very interesting podcast ahead for us. You're good? Everything good with you? Anything going on?
Just, got the new M5 that's 17 years old that you've assured me all the lines are going to cost me $20,000.
Good luck with that.
Okay.
Let's bring in our guests. It's Dr. Jill Weimer and Chris Velona. Dr. Weimer works at the Stanford Research, not-for-profit research organization, where she's focusing on understanding the processes that control development of the cerebral cortex, which I am fascinated by. And Chris Velona is the father of a son with a genetic disorder we're going to be talking about. First, let me welcome Dr. Weimer. Welcome.
Thank you for having me.
And then Chris, are you there?
Yes, hello.
All right,
Thank
Chris, you sit tight for a second while I mine Dr. Weimer for just a second.
Of course.
Before we get into Batten's disease and genetic disorders, talk to me, Dr. Weimer, about your work generally.
Sure. So I would say our lab kind of bridges from the bench to the bedside. So we have projects that are very basic in nature, understanding how stem cells divide in the developing cerebral cortex, how they migrate to where they need to go, how they send out axons to make those long-distance connections that we rely on for learning and memory and motor movement. And then we kind of take that to the next level and understand when genes that are important for that process are mutated, how does that lead to different developmental disorders, such as Batten's disease that we're going to talk about today. And then sort of the third arm of the lab is to really look at preclinical therapeutic development. So how can we test various drugs or different types of treatments that then could help those maladies? Anything
particular you guys are excited about right now?
Actually, we have a very exciting arm in the lab where we've been developing genetically modified pig models of disease because we're finding that with some of the diseases we study, most of the work we do is in mice. And when we mutate the genes in mice, they don't form a human disease or a human-like disease. So we've actually moved some of our modeling into pigs, which is pretty exciting.
So pigs are really much closer to human genome than other animals, right?
Yeah, and the pig models we work with are they're a miniature pig, swine, but it still gets up to about 200 pounds. So the organ, the body size, and metabolism is much closer to a human man. So I think we can learn a lot more from them. They're a lot more expensive to maintain and a lot more difficult to work with, particularly for somebody who does a lot of neurobehavior testing. Trying to get them to run through mazes is a lot harder than training a mouse to run through a maze, but I think it's a challenge that we really enjoy.
I am very interested in the cerebral cortex. Dr. Weimer, give us a little primer on the different layers of the cerebral cortex, just so the listeners, because we got to sort of translate this for the average person. And what we're talking about is cerebral cortex, the layers, and what makes our cerebral cortex different from every other.
Sure. So what we're talking about is that there are these different layers, and each of the different layers connects to different parts of the brain. So some layers might connect within the same hemisphere of the brain, so the same side, your left or right side. And each portion of the brain, so the front, the middle, the back of our brain, each control different functions. So the front might control higher order processing. The sides of our brain control hearing. The top of our brain might control motor movement. The back of our brain, surprisingly, is where our vision is processed. So kind of within those different layers, they're going to connect to different portions and control different things. And unlike mice who their brains are much smaller, there's many fewer neurons that are packed into that space. We also have something unique that with these layers kind of come these folds, these what we call sulci and gyri that kind of give us more surface area, and we can pack a lot more neurons into a smaller space. So the pigs actually then are much closer to humans, that they start to get some of the same sulci and gyri and the same folding that we have in the human brain.
We have how many layers in the cerebral cortex?
We have 6 layers in our cerebral cortex.
And the one layer that we have that the other great apes do not have is the pyramidal cells. Is that right?
Well, so you actually have pyramidal cells in different parts of your brain, and they have pyramidal cells. It depends on which area of the brain that you're looking
I
in
was
that
studying.
will have different
I was studying something once where I thought, oh, this is really interesting. It was sort of a laterally. It was a cell type that other primates didn't seem to have or didn't have in the
and I don't know which one it would be. There's depending on if you're looking in auditory cortex, there's different, even within humans, sometimes you'll reduce down to a fewer number of layers as you move into the cingulate gyrus.
Mm-hmm.
It
just seems that we integrate the structures
better.
And we have more of an integrative, and that's really why we can come up with new ideas and new learning and all this crazy stuff. And you're actually studying. Can you summarize that in a kind of couple sentences for us? What that is, and how that works?
So in the last couple of years, a lot of what our lab has focused on for the basic neuroscience is actually how we actually, once an axon's formed, how we actually traffic and communicate stuff down that axon. So we have axons that go great distances. Probably the longest one in our body goes from the tip of our spinal cord down to the tip of our toe in our sciatic nerve. And so unlike other cells in your body where all of the nutrients and all the things that are happening in the cell really happen around the nucleus, in neurons, we actually, as they grow and form and over our lifetime, we have to maintain those nutrients. So maybe mitochondria for energy or packaging of neurotransmitters for synaptic transmission have to go down these axons. So our lab's been looking at some of the mechanisms that actually move or traffic that cargo. I like to liken it that say you wanted to ship some apples from LA to Albuquerque. And so first you need to have a signal that tells you need apples in Albuquerque. Then you need to look in all those train cars that are sitting there in the train depot and figure out which one has apples on it. And so in the neurons, what we're doing is studying those mechanisms that kind of are almost like the barcode readers that are figuring out that I need apples and then you don't accidentally send oranges to Albuquerque.
Is there are these regulator genes we're looking at mostly, or what kind?
Yep. So we think that we have sort of some key
scaffolding
proteins,
the Fox,
and with
FOXP2 and all that business.
Yep. And actually, it kind of then bridges into these Batten's disease genes. So
all right, let's do that. Let's get there.
For CLN8, which is one of the forms of Batten's disease that Chris's son has, and then another form that we've been working on, CLN6, we actually think that these proteins form almost a barcode on the vesicles, so that these proteins can kind of come in and read that barcode. And we don't know what the content is that's in the vesicles that CLN8 or CLN6 are labeling, but we think that it's important. And if you lose that barcode, then that cargo doesn't make it to the nerve terminal.
When you say vesicles, we're talking about the neurotransmitter vesicles?
Yep. They could be. We don't know what's in them right now. So it could be vesicles. Neurons are very unique that we build proteins even at the nerve terminal. So it could be some of the building blocks that we need to make synthesize those proteins. It could be enzymes that are important for modifying those proteins after they're made. We're not quite sure. So that's one of the areas that the lab's really heavily focused on right now, is figuring out what the cargo is.
So that was all Dr. Kaplan's work in the periphery, of the nerve, right?
Yep.
So there are messenger RNAs out there in the periphery of the synaptic button that allow. They're actually functioning. They build proteins out there and modulate too, and regulate it.
Yep. And so one of the possibilities is that the cargo that's being transported is these messenger RNAs.
Oh, interesting. Oh man, would that be a breakthrough?
Yeah.
That would be crazy.
Yep. So it's an exciting field. It's an exciting time to be a neuroscientist.
Oh my God, my mind just boggles with that because it has so many implications. But anyway, so let's talk about what Batten's disease is, and then we'll get Chris to tell his story in just a second. But clinically, what is Batten's disease?
Sure. So Batten's disease actually is a term that we loosely use to describe about 14 different diseases. They're collectively put together because of the brain pathology that we see, so the changes in the cells in the brain, and then also the phenotype. So the form that we're going to talk about today has sort of a a late infantile onset. So the kids usually are perfectly fine until they hit their 3rd, 4th, 5th birthday. And then depending on the form, the symptoms can vary on what you see first, but it's usually motor coordination deficit. So these kids have been completely fine. They might start to have some stumbling. You don't think anything of it at first, and then maybe they start to have seizures. A lot of the kids will then start to have visual deficits, and it just all goes downhill from there. And it can go downhill very, very rapidly. And these kids with the CLN6 form usually pass away by their 10th to 12th birthday.
Wow. CLN6, is that what you said?
Correct. Yeah. Yep. So the disease is called, we collectively call it Batten's disease, but and it was one of the physicians that first described it around the late 1800s was last name Batten. But it actually, there's another name that we use to describe it too. It's neuronal ceroid lipofuscinosis. And ceroid and lipofuscin mean that the neurons are accumulating this storage material, this ceroid material. And it's something that you see in Alzheimer's patients. As the patients get older and older, they start to accumulate this junk in their lysosomes. And the kids with Batten's disease, they prematurely accumulate that storage material.
Now, I would think that would kill off a bunch of neurons, right? Is that what's going on?
Yeah. Well, we actually, just like with many neurodegenerative diseases like Alzheimer's, like Parkinson's, Huntington's, they accumulate junk in their cells. We don't know if it's actually what's killing the cells or if it's the cell's way to try and protect themselves.
Once the cells are gone, can anything be done?
As of right now, no. So there's a lot of folks doing research on neuronal regeneration, trying to restore neurons, using stem cells to try and get those neurons that have died. But once they're gone, they're gone.
So then the
goal
is to prevent
with Batten's disease.
Yeah, prevent degeneration. So what can be done to prevent or to slow or whatever? Go ahead.
So some of the... Oh, go ahead.
Yeah.
Yeah. So that's what I was saying is that with the kids, that's why, as we start thinking about treatments, it's really, really important for us to be able to diagnose the kids as early as possible so that when we do have effective treatments that we can get those into the kids as quickly as possible so that there are as few neurons that have died as possible.
So you know the exact chromosome where the defect is for each of these?
Correct.
And so why can't you get the right chromosome, put it on a virus, and
stick it in
shoot it in there? Like give them an adenovirus, give them a cold.
Or clip this one out.
Yep. So that's actually, I think, what we're headed towards. So I'll tell you sort of a story that's this is how Chris and I got connected. In August of 2015, I was connected with a family based out of LA area, and their daughters had recently been diagnosed with CLN6 Batten's disease.
Two daughters, two of them. Oh boy.
Yep. So, and this is a Hollywood movie producer by the name of Gordon Gray. And Gordon, they were diagnosed in March, and it took him from March till August for him and his wife, Kristen, to connect with me, just because even when they went in and they were seeing the physicians, they went around and around for months of not knowing what it was. And even though we know what the chromosomal location is, we know what the gene is, they were misdiagnosed early on with, your oldest child, Charlotte, the one that was showing the symptoms of the disease. Oh, this is, she has mild autism. She has
muscular
this, she has
dystrophy
this.
Or something.
Wow.
Yep. And so they went around in circles for quite a while. And then finally in late March, early April, they finally had whole exome sequencing and figured out that it was Batten's disease. And so Gordon and Kristen started calling everybody that they possibly could that studied Batten's disease, connected with the right people. And I would actually say we first connected in July. I met Kristen in July. But what happened in August was they said, we're going to bring together this group of scientists. We're going to tackle this disease, CLN6, head on. We're not going to take no for an answer. We're not going to sleep. We're going to fight, and we're going to find a treatment for our daughters. In early August of that year, through some connections that we had put together for the scientific board for their foundation, I was connected with a scientist at Nationwide Children's in Columbus, Ohio, by the name of Brian Kaspar. And Brian's lab had done just what you were saying. They had taken the gene for another rare pediatric disease called spinal muscular atrophy. They had loaded that into a common cold virus, adeno-associated virus, and they had used that to introduce it into children with spinal muscular atrophy. And SMA is also referred to as floppy baby syndrome because kids with the more devastating form of SMA have muscle problems from birth. They can't sit up. And these kids are usually, with the severe form, dead within a few years.
I had a patient with spinal muscular atrophy, and he lived well into his middle ages, and he observed something interesting about himself and everybody else with the condition, is that they became smarter as they aged. His cognitive abilities expanded dramatically, and he was an accountant. So he could specifically measure, sort of see what he was doing.
Why?
That's why I brought it up. But you're
That's
court.
insane.
Yeah, it's insane, right? Maybe all those integrative functions are somehow enhanced. Yeah, isn't that weird?
Wow.
Yeah.
But look that one up. See if
Fascinating.
I think SMA4, right, is the milder one. Is that right?
Yep.
Yeah, I think he had SMA4 if I
Yep.
remember right. And it's a long time ago. But he really was super smart. But in a nursing home by the end. Yeah.
Wow.
It's crazy. It's crazy.
Yeah, so they were really successful with this, and so we said, could we model the same thing with CLN6? And so normally, as you know, if you're going to develop a drug, any sort of treatment, you're very sequential in how you do that. So we develop the gene therapy, we put it into a mouse model, we test it, we make sure that it's safe in the mice, and that can take a couple years. In fact, I still have from the very first mice that we have mice that are that we treated in probably October of 2015 that are still alive, right? So this is a very long process. And only
that's just the animal model part.
Yep, that's just the mice, because then usually your next step is you have to go into some larger animal, dogs or monkeys, and do toxicology studies, show that it's going to be safe, and then only at that point do you actually make the human-ready drug or the GMP manufactured drug, because that's probably the most expensive step before you would actually go into a clinical trial. So that whole process in drug development usually is anywhere from 5 to 7 years. And the Greys knew that if they followed that same time course, that their daughters would be dead by the time we got to an effective gene therapy.
And let me ask, is somebody following that path presently?
Yep. So we started in August of that year. We started making the virus. We injected the first animals. There's a bunch of regulatory steps that you have to go through for any drug approval. But essentially, with the Greys' help and partnering very close with them and their scientific team, they said, scientists, the reason you go sequentially like this is because there's a lot of risk that goes along with this, and there's limited funding to do your science, and so we're going to remove that risk off your shoulders. And we want all of these things to happen simultaneously. So you're going to go into, as soon as the Weimer lab's injecting mice, the Caspar lab's going to be doing the non-human primate studies. And we're going to go into GMP manufacturing right away as quickly as we can get into a facility to start making the drug. So we were actually making the human-ready drug before I had any data from the mice.
So is it a fast-track process through the FDA?
Yep.
Okay.
So we went to the FDA with our IND application in January. We did our pre-IND. Our full package went in February, and we got FDA approval to start a clinical trial in March of 2016.
With these two girls, or more than that?
There were 6 patients in that first clinical trial. That actually, and the clinical trial is being done at Nationwide Children under the direction of Dr. Jerry Mandel. They've actually enrolled all 6 patients. And so what we've been working on now, what the Nationwide team's been working on now, is sort of what the next step is beyond those 6 patients, whether we go back to the FDA and look at possibly expanding the number of patients that we're doing in that phase 1 safety trial, or kind of taking this to the next step.
How did it work?
We actually can't say because it's still an ongoing clinical trial. But it's, I would say, probably looking very promising if we're looking to expand it.
And if you expand it, how could somebody get into that trial?
So in the first phase, in phase one, essentially the kids had to meet certain criteria. So you have to the clinical team at Nationwide has to really spell out for the FDA how they're going to define the subjects and how they're going to be monitored in a safety trial. And so essentially the kids would have to come in. They see a peds neurologist, Dr. Emily De La Reyes at Nationwide, and she evaluates them and kind of determines on a rating scale where their disease is at that moment. They also get a lot of MRIs, neurological scanning to look and see sort of where their brain is at that time. And so in that first phase, the kids had to meet certain criteria. So they had to be kind of earlier in the disease progression. And one of the things that they could propose if Nationwide decides to go down that route is to kind of expand where the kids could be in the disease progression for the treatment. Now, the thing that made it a little hard, and actually even as we start to talk about the form of Batten's disease that Chris's son Sebastian has, CLN8, when we started this, in addition to the two gray girls, I knew of two other kids in the United States that had CLN6. So it's that rare. But because of the awareness that the Gray Foundation was able to bring to Batten's disease, we now know of at least 18 kids in the United States that have clinically diagnosed CLN6 Batten's disease, and I know of at least 30 outside the United States. So it's a growing number, a growing pool of kids, and it's great because now there could be a potential treatment for them on the horizon.
Let's take a quick break, and then we will flip into Sebastian's story. Well, one of the great parts about working in recovery is seeing former patients successfully move on. And I've had patients that have come up to me years later and shake my hand and say, you know what, sometimes people are kicked out of treatment. And many of these folks move on to become mental health professionals themselves. And, of course, the field of psychology is vast. The need for competent practitioners is huge. If you're considering this rewarding career, I urge you to consider the California School of Professional Psychology at Alliant University. Now, I've known them for a long time at Alliant University. I've spoken at their past events. It was founded in 1969. It's boasts an alumni network of nearly 50,000 people worldwide. And Alliant has fostered many of today's mental health pioneers, authors, and advocates. CSPP at Alliant University hosts both on-ground and online programs in business psychology, marriage and family therapy, clinical counseling. They also offer APA-accredited doctoral programs in clinical psychology that can allow for specialization in child psychology, clinical forensic psychology, and integrated psychology. And the faculty is crazy. It's made up of leaders and historical figures like Abraham Maslow, Carl Rogers, Viktor Frankl, some of the true fathers of modern psychology. For more information, and I worked alongside of these students as well, by the way, in the clinical setting, as well as having lectured at the institution. So for more on the California School of Professional Psychology, CSPP, at Alliant, click the Alliant banner on our website or visit Alliant, ALLIANT.edu, alliant.edu. All right, we're back. Weekly Infusion. Bruce, interesting stuff, right? This is great stuff. Chris, are you there?
Yes, Dr. Drew, I'm here.
Hey, man. So let's hear the story of how your son Sebastian presented, and we'll again walk through this, give it back to Dr. Weimer, and talk about what Canada needs to be done.
All right. Well, great. Well, thank you so much for having me on. I appreciate you very much, Dr. Drew. Known you for quite some time, and I do appreciate you doing this for me and my family. So, kind of like a little bit like the Gray story, like Jill was telling everybody, you know, for a couple of years, more so like 4, Sebastian was misdiagnosed, and it started out with epilepsy around the ages of 4.5 to 5 years old, roughly in the fall of 2007, and didn't think anything of it. Then he had another seizure, and then another. So then we, checked him into a doctor who put him on some epileptic drugs, and then, of course, trying to find the right drugs for years. And then the progression of this disease, unbeknownst to us, just kept unfolding to where it wasn't just epilepsy. It was more like he couldn't talk normally. He was tripping a lot. His cognitive functions were slowing down. He was having issues with speaking. And, we noticed that there was a lot of different things going on, not just epilepsy. Of course, the seizures were still there, but this just wasn't normal for epilepsy. And the people at who were treating him at UCLA said, maybe we ought to test him for something else. And that's when we decided to do genetics testing in the fall of 2012. And we found out that he was diagnosed with Batten's disease of the variant of CNL8. So, of course, it's devastating. And sorry.
Right.
But, what do? You just try to find anything you can to move forward and try to, figure out a solution, for him, and so from 2013 to 2016, we're just kind of like, what do you do? And like Jill had said, there's not a whole lot of stuff going on out there, So what we did was I decided that I started the Project Sebastian for epilepsy and neurological disorders to help children back when he just had epilepsy. And through that, I started just combing the Facebook pages and internet, and started putting in hashtags for Batten's disease. And I had heard about the Charlotte and Gwyneth Gray Foundation through my ex-wife, and she had told me that she had heard about these people. And so, I just Googled them, and I picked up the phone and I called their office. And within 2 days, I was on the phone with Kristen Gray. It was really cool, because she just picked up the phone and she goes, "Hey, so I hear you're trying to get a hold of me. What's up?" And that's how she is. She was just very amazing and just real as it can be. And she heard my story about Sebastian, and she goes, "Yeah. I think we should have a meeting. That would be great," because I really didn't know, and I didn't know how to do it, and I hadn't met anyone that had a child that was like Sebastian. So we ended up having this lunch. Actually, in the fall of 2016, after that first conversation in late summer, and Kristen and Gordon met with myself and my ex-wife and my son Sebastian, and just told us everything that they had gone through and what they were doing. And we asked them, begging them basically, "Hey, can we get in? Can we get in on this? Is there something we can do?" And they were very, very happy to say yes. And they just told us that it's a long road. It's a very the gene therapy was something that was brand new, that had been proven in other types of neurological illnesses and stuff like that, and that they had started some CNL6 trials. And even though Sebastian had 8, that they thought that it might be something that could be workable. So they introduced us to this amazing team, one of which is Jill, which is on the phone. And that's how it started, and it's been amazing ever since. So right now we're in the beginning stages, as Jill was explaining. So we're very hopeful, and pretty much that's it. Every day is a grind, and what we're trying to do is we're trying to raise a lot of money for the procedures, because unfortunately it's not covered by any insurance. And it's very expensive. It's in the millions of dollars. So we're locally and nationally trying to fundraise, and curebatten.org, which is the Charlotte and Gwyneth Gray Foundation, have been very helpful in helping us put together this team, and started doing some of the research as well.
Is there somewhere people can go if they want to support?
Sure. For Project Sebastian, to find out more about Sebastian's story and to see what upcoming events are for fundraising ideas, or just to donate, you can just go to www.projectsebastian.org. And basically, those fundraising dollars will benefit Sebastian's cause through the Gray Foundation as well.
So, Dr. Weimer, does the fact that he is CNL8 change the protocols he has access to?
So we actually, it's kind of like we're starting at the beginning, but again, kind of applying this accelerated pace that we use for CLN6.
Yeah.
So the idea is, of course, we have to put the CLN8 gene back into Sebastian and the kids with CLN8. We couldn't use the CLN6. We are with the Grays, in addition to the gene therapy. When they said go in August of 2015, they were saying go to many different types of treatments because they didn't want to put all of their eggs in one basket because they didn't know if the gene therapy was going to be successful. The gene therapy is what's really customized for the particular gene, but a lot of the work that we're doing kind of outside the gene therapy space, so looking at small molecules, repurposed FDA approved drugs, different pharmaceuticals, different stem cell applications, those actually would be wide reaching and could have benefit in all forms of Batten's disease. So I think even beyond the gene therapy that we're starting for CLN8, there's a lot of other work that we're doing too that also could have benefit to the CLN8 patients.
When Chris first told me about the story, my question was, how come this research is not being funded by the NIH or any of the usual funding organizations?
Yeah, so I actually have funding from the NIH for CLN6 work that focuses kind of on the basic questions. The problem is that the NIH doesn't have sort of this fast track mechanism that the Gray Foundation, the Cure Batten Foundation, and Project Sebastian are working towards. So they do really rigorously want you to follow sequential order.
So if you apply to the NIH, you'd be doing the mice research, period.
Yep, yeah. Correct. Yep, and then you do the next step, and then the next step, I think is logical in some cases, but in these rare diseases where the kids don't have that time, there has to be a new model.
Can you go get the money for the NIH mice studies while you try to raise money for the other two?
Well, and if you're looking at NIH dollars, even the time course for us to secure an NIH grant is usually about a year, even just to apply for and get an NIH grant funded. So, with the Grays, we said we don't have time even to do that, right? Like this has to start tomorrow. And we can't sit around and wait on a review panel to look at this. And so that's why I do think there still needs to be rigorous scientific oversight in the work that we're doing. Yep. So, Chris and I met yesterday about a scientific advisory board for Project Sebastian. And then also even partnering, there's a couple of other CLN8 foundations. So there's a family, the Scarpetta family in Rockford, Illinois, that Chris has been working closely with. They have a son named Brock who has CLN8 Batten's disease. And so how do we partner those groups together since they all have a common goal? And how do we get everybody synergized together and then give it the scientific rigor so that we can work our tails off, make sure we get this done as quickly as possible, but we're doing it right.
And Jill, your attitude is amazing given this is hair-raising stuff. This is so I'm not a pediatrician. I can't handle it. I really can't. Yeah. I did not like doing pediatrics. It was too much for me.
It's painful to see, and go through the process. And with the Grays, like it is, the first probably 6 to 8 months, half the time that I would call Kristen, and we were probably on the phone almost every single day with the Grays. She'd say, "Here, talk to Charlotte. Talk to Charlotte, right?" And see, so you can see firsthand like what's going on, and it really does push you. It pushes you really hard, and I have to say that the way that my team and the Caspar team at Nationwide really stepped up to this was unbelievable. We joke in my lab that now that we've kind of crossed the finish line with getting CLN6 to the clinical trial, that there's no turning back, there's no slowing down, and so we're tackling everything head on, and we really want to kind of just keep rolling through these.
I don't know the sort of literature, the landscape of literature for gene splicing. We're doing gene splicing here, right? Gene therapy.
No, this is actually gene therapy.
Gene therapy. For neurological disorders, but I would imagine this is going to push things forward for a lot of things, no?
Yep. And actually, even just I would say since we moved this forward, there's been a lot of movement in other forms of Batten's disease. So the probably the largest Batten's disease foundation, the Batten's Disease Support and Research Association, the BDSRA, is holding their annual family conference right now. And I know that during that conference, there's a number of sessions that are kind of going through, here's the landscape for what forms of gene therapy have already been launched. Here's the ones that are on the horizon. And I think it's just pushing not just private foundations and academic institutes, but even pharmaceutical companies that are invested in gene therapy to say, hey, we got to move faster because the clock is ticking. we got to get these things going a little bit faster.
Isn't there is there any centralized process? There's so many genetic disorders and compassionate care projects pushing the FDA ahead more rapidly.
Yeah.
But is there a centralized? It would seem like everybody could get together and.
Yep, and surprisingly, a lot of times it does fall to these family foundations.
Yeah.
They're probably the biggest advocates. So there's a family out of Illinois, Tracy Van Houten, and this man he has 2 kids with CLN2 Batten's disease, and he, from the time that his son Noah was diagnosed, he has spent countless hours, badgering the FDA. This has got to move faster. These things have got to move faster. Going to Washington, D.C., lobbying senators, invest more money in rare diseases. He's been probably one of the most amazing advocates for Batten's disease collectively. And they finally just actually got an enzyme replacement. So CLN2 is a little bit different. It's an enzyme, a protein modifying enzyme. And so that one they were able to actually work with Weimer to come up with a enzyme replacement therapy that just received FDA approval about 2 months ago. Now, for Tracy's family, very unfortunate. His son Noah passed away just under a year ago. So he fought the fight, and Tracy knew he was fighting this fight for all kids with Batten's disease. And I think Chris and the Grays are the same. They know they're fighting for their kids, but they're fighting for these other kids that have these this horrible disease as well.
Yeah, absolutely. I can't thank enough. Jill has been amazing in helping me understand the scientific side. But as a parent, touching base with these other families across the country and across the pond, it's been amazing. It is singly just a race, and it's the question that Dr. Drew had asked earlier, where can't we get the funding? Where's the funding coming from? It's coming, but right now, if you waited for that funding, unfortunately, your child would deteriorate and pass away. So it is a race, and that's the thing that really sucks about this thing, is like creating awareness is everything, because when you create awareness for something, then you create the dollars that can go into the research and to possibly save some of these children's lives.
And I just keep thinking how this could push other, you don't even know the ripple effect in other scientific endeavors and other disease states and things, that this is cutting edge, this is state of the art.
Yep.
And let me just ask kind of a weeds question. The difference in the different CNL types, does the CNL stand for something, or just sort of a random nomenclature?
It actually was the nomenclature when I said that the name collectively is neuronal serine lipofuscinosis. So they took the NCL, and gave it the CLN.
And then each of the numbers is a different gene. Different
correct.
Gene, with the lipofuscin accumulation. That's the downstream effect. Is there a common physiology here?
Usually, most of the forms, the kids have visual deficits, seizures, motor coordination, and cognitive impairment. There are a couple of adult forms, so Kuss disease.
I'm asking a little different. Is there a common biochemistry, I guess, is what I'm asking.
Oh, no.
Yeah.
So actually, there is not. So the CLN1 and CLN2 and CLN10, which is also called cathepsin D, those are all lysosomal enzymes. But aside from those three, there really is not any common function between the proteins.
Isn't that interesting?
So are these all lysosomal storage diseases?
Yeah, it's a lysosomal storage disorder, but not all the proteins are even lysosomal localized.
Isn't
so CLN8 and CLN6 are ER, endoplasmic reticulum proteins.
Now this stuff, isn't this goes on in normal aging too?
Yes. So normally, like the older we get, I think probably, the neurons in our brain are born before we are born, before we hatch. And so I think just over time, as the cells start to slow down, they accumulate some of this junk with normal aging. In neurological disorders like Alzheimer's disease, you accelerate that accumulation. And then these kids with Batten's disease just have it at a much, much accelerated pace.
Do you have a theory of what that biochemistry might be? If it's the endoplasmic reticulum, it's something to do with protein. What protein? Putting together proteins.
So one of the philosophies is that, with all of these neurodegenerative diseases and Alzheimer's disease, you have accumulation of these extracellular amyloid plaques, and in the cells, this hyperphosphorylated tau. And one of the ideas is that the cells just kind of start to slowly, the lysosome starts to slow down its function, and so it's the cell's way of kind of protecting itself. It sequesters this stuff in one spot. But I don't know, because even in Batten disease, people have actually tried to take that storage material and do some protein biochemistry, bust it apart, and see what is in that material. And even between the different forms of Batten disease, the different proteins that accumulate are different.
Wow.
I just got a feeling it's something to do with protein synthesis, right? It's got to be
or protein degradation.
Yeah, some protein metabolism byproduct sort of thing.
Yeah.
Well, listen, you guys. Chris, I want to give you a chance to put out another plea, if you don't mind. And how's Sebastian doing today?
Sure. He's playing video games. So
good.
he's good today.
Good.
Every day is different. So I'm just grateful that we have one more day with him, and to keep grinding and to keep raising awareness. What I'm trying to do now is I'm getting crazy with raising some awareness. So this coming fall, in August 19th, I'm going to be climbing a 14,000-foot mountain, Grace Peak, with some other Batten's disease families to raise awareness and dollars in Colorado.
Which is easy compared to what these researchers and these kids are going through.
Yeah, I mean, I'm trying to do my part. I'm going to get into incredibly stupid shape for this stuff, so I'm going to be doing a lot of different things along with this climb. You can go to crowdrise.com to search that for the Charlotte and Gwen Foundation for that on that particular, and you can just raise money there too. I also have many different outlets. Of course, I got the Facebook, Instagram, Twitter, GoFundMe, all these different areas that you could possibly donate. Also, we're doing a spin for Sebi, a local challenge here for stationary bikes that's coming up. And like I said, I appreciate the kudos. You can always go to projectsebastian.org to find out more. And if you also wanted to check out how this all started for me and my journey, you can go to curebatten.org as well. That's the Gray's family website. So thank you,
guys. Well, listen, Jill, congratulations on this research. This is phenomenal stuff.
Yes.
Thanks. And thanks really helping get the word out for us. We really appreciate it.
We appreciate it.
How'd you end up in Sioux Falls?
Actually, so I was doing my postdoctoral. I did my PhD in Rochester, New York, and I had was doing my fellowship in Chapel Hill, North Carolina. My PhD advisor from Rochester calls me and says, "Hey, I'm starting a new research center. You and I work together great. You want to come and be my first faculty recruit?" And I was like, "Sure, where are we going?" And he said, "Sioux Falls, South Dakota." And I said, "You're British. Do you even know where Sioux Falls, South Dakota is?" But I went out to visit, and they had actually just built this amazing new research institute there, and they're doing great stuff. They had a very heavy focus on pediatric research, and we now have 4 different labs that are working on Batten's disease there. So it's grown tremendously in the last 8 years since I started there.
Great. Well, congratulations on all this, you guys. We appreciate you sharing this with us, and we will, we'll check in on you, Chris. I really hope that things turn out okay, and if not, you will have done a great service for those following our generations, let alone these kids. So appreciate it.
Thank you very much for having me, and thank you, Jill, for joining us, and I appreciate it. I really do.
No problem.
All right.
Thank
you guys.
Stuff.
So, Bruce, pretty interesting stuff, right? I feel terrible for Chris and Sebastian and all that. I mean, can you imagine?
Yeah, but it's amazing. These are diseases with no hope, even recently.
I know.
Now sounds, more hopeful. Pretty hopeful.
It's so
fascinating science behind
such
it.
high stakes game here, and just hats off to Weimer, Dr. Weimer, and her team. That is a serious. Those are serious scientists.
Yeah.
I mean, she was giving us the sort of really the layman's version of her work. Trust me. Even if it seemed difficult to understand, if you really got into it with her, believe me, it would be hard to understand. Right. I mean, they're working in rarefied air, but God bless them. I'll tell you what. All right, so we have a bunch of emails. I don't really have time to get to them. Just here's a quick one. My husband has been an alcoholic for 30 years. Recently stopped drinking with help from Kaiser. Now that well done, Bruce. Now that he's stopped drinking, he's become very emotional and constantly feels like crying. Is that common for someone to break down like he does? Yes.
Is it 90 days?
Yeah, well, even the first 6 months, people can have all kinds. But the program of recovery is designed to manage this. Any alcoholic that stops drinking or drug addict stops using that doesn't do 12-step work, do the steps, is going to feel like hell and going to engage in behaviors and feelings that are going to be problematic. The program is designed to deal with the anxiety and the depression associated with abstinence. And of course, people feel differently.
But I think that's one of the reasons for 12-step, because you continually have people that say, well, I can do it on my own. I can do it with nothing.
You can do it, but you will not be good.
I think it anticipates some of the emotional baggage or trauma that comes out.
That's right. But it changes over the first year quite a bit. And, that's why people revisit their inventories and revisit their third steps and revisit these things, because they have different meanings at different times, but you have to do it. You have to do the work, or it doesn't stick. It doesn't stay. Now I used to always say, no sponsor, no recovery, no treatment. And so you got to get sponsor, you got to get steps, you got to do your thing. And some of that emotionality will, if not settling down, it'll sort of make more sense to him, because he'll realize that he is not alone. So there you go, Sarah. All right, let's see, we've got another one in here. Here's somebody sleeps with their jaw and hands clenched. I wake up in pain. Would love to know how to make this stop. Stress, the doctor's telling him. I hope that's all it is. Sounds like it could sleep disturbance, seizure. I'd worry about many other things, but it can just be stress related, right?
Most likely.
Yeah. My doctor says sleeping, blah, blah, blah. I wear a teeth guard at night, not grinding her teeth, just clenching her jaw and her fists. Not much can be done with that, unfortunately. Try melatonin, maybe? I know it's going to do much for sleeplessness. That's more sleeplessness or chronology of sleep, how sleep timing goes.
Grinding
maybe more exercise?
They're on medication or sometimes side effective. But, that's a good one.
Maybe more exercise during the day, perhaps? That's a hard one. Maybe what you might do with her is get her a sleep study and see what really is going on. Maybe there's all kinds of other kinds of movement problems during the night that can mitigate some of the stuff that she's experiencing.
Right. Certain diabetes, blood sugar.
We'll get a full medical eval for sure, but I'm saying get a formal sleep evaluation where she's observed on EEG and monitoring during the night, so we can see what this, get a name for what this thing actually is. All right, let's wrap this up. We got lots more messages and things. We appreciate you guys sending them to us at doctor.com/contact. Also, our friends at HEAL, don't forget those guys, and the Alliant University and the CSPP. That's a great training ground over there. And again, HEAL. Why would you do anything else?
Stay away from my ER.
It makes no sense to me. Yeah, stay away from his ER. Go to HEAL.com. Get the HEAL app. Have somebody in your house within 2 hours, and that's that. And it's way less expensive than going to anybody's ER, anybody's urgent care. Like literally a tenth or less than that, no matter what.
I had somebody with an ER copay last night, $300.
Yeah.
Not happy.
Yeah, I'm sure they're not happy. All right, we'll see you next time.
Remember, you can find all these podcasts at drdrew.com. The Dr. Drew podcast, the This Life podcast with Dr. Drew and Bob Forrest, and the Adam and Drew podcast, which is available 5 days a week. Find them all on iTunes and rate us 5 stars. Subscribe and get it first. And if you're really happy, click on the Amazon banner at drdrew.com to help support the show. We'll thank you for it. If you join the email list via drdrew.com/contact, we'll send you a weekly infusion newsletter with Dr. Drew's news. We're so grateful when you get in touch. We read all your emails and we'll bring you the subject matter you want to hear about.